today we are starting our new unit beginning with blood. We have here your level 1 objectives listed. They include things like listing the functions of blood, the percentage of each of the following that contribute to the overall volume of blood, being plasma, buffy coat, and erythrocytes. So I want to know what percentage of plasma contributes to the overall volume of blood, what percentage of the buffy coat contributes to the overall volume of blood, and we'll talk about that in a little bit. I want you to list the 3 major components of plasma, list the 3 formed elements, define those terms that you see there, hematocrit, antigen, antibody, and so on. Describe the difference between granulocytes and agranulocytes, and then list the 5 types of leukocytes. And for our last level 1 objective, describe the anatomy and function of a platelet. Level 2, a little bit more detailed, describe the functions of each plasma protein and list them from most to least abundant. Describe the anatomy of erythrocyte and hemoglobin, and then describe the anatomy and function of each leukocyte. For our level 3, describe the life cycle of erythrocyte. We're going to talk about antigens and antibodies and how they determine our blood type. And then we should be able to describe the results of a blood transfusion for various scenarios. All right, let's begin with blood. Blood is a fluid connective tissue, as you already know. Because it is a connective tissue, it contains cells, proteins, and a ground substance we refer to as plasma. On average, blood is kept 1 degree higher than body temperature, so instead of being at 37 degrees Celsius, it's kept at 38 degrees Celsius. And this contributes to one of the functions of blood. Now, blood has many functions, the first and foremost being transportation of oxygen and carbon dioxide. But blood also transports things like enzymes, hormones, as well as nutrients and waste to and from the cell. It also helps regulate multiple things, including body temperature. As I had mentioned, the blood is kept 1 degree higher than average body temperature. That is because as it moves away from the core, it is releasing that heat and warming up the internal organs as well as the appendicular region, the limbs, if you will. Throughout this semester so far, we have learned about different fluids in the body, including CSF and synovial fluid. All of these, as I've said multiple times, are modified versions of blood plasma. So essentially, if we can control the fluid volume of blood, we have the potential to control fluid volume all over the body, regardless of what fluid it is. The last thing blood helps us to regulate is pH. pH, many of you know it as the pH scale, is a measure of hydrogen ions in a solution, and that number of hydrogen ions tells us if our solution is acidic or basic. So if we look here, what I've done is drawn the pH scale out for you. It's usually measured from 0 to 14, 14 being basic and 0 being acidic. Now, our blood contains something called bicarbonate. And bicarbonate is what we call a chemical buffer. Because what it does is it attaches itself to hydrogen and it becomes carbonic acid. And carbonic acid can become CO2 and water. And in doing so, it allows us to breathe out the CO2. Right? If you think about it, when we breathe in oxygen and we breathe out CO2. So if we can essentially get rid of the CO2, that means that we can no longer go this way in our equation because we're missing one of the ingredients, meaning that we've reduced the ability to have this hydrogen here affecting our pH. So let me explain that again. The amount of hydrogen in a solution affects the acidity. So, if the amount of hydrogen goes up, then that means that the acidity also goes up. And we say that our pH then goes down. It's closer to 0. At the same time, if we reduce the amount of hydrogen in our body, we are reducing the acidity of the solution, meaning that our pH goes closer to 14. So again, this bicarbonate in our blood is able to bind itself to hydrogen, kind of masking it in the solution. And eventually, we're able to break apart this carbonic acid formed by this bicarbonate and hydrogen binding into CO2 and water and breathe out the CO2. When we breathe out the CO2, we're left with water, which means we cannot become carbonic acid, which means that this hydrogen is no longer free floating. It's in that water, and as you know, water is not harmful for us, it's actually good. So we've reduced the acidity of the blood by having this bicarbonate there. So the bicarbonate in the blood helps us to regulate pH by binding to hydrogen when we become too acidic, allowing us to breathe it out. The other function of blood is protection. We have leukocytes in our blood that allow for an immune response, and these leukocytes allow us to produce antibodies, and that's something we'll explore a little bit more when we get to the lymphatic system. Alright, your book talks about blood in the following manner. They show blood being drawn from an arm and then put into a test tube and then centrifuged, basically spun around very, very quickly. And what this does is it separates the components of blood into three parts. We have plasma, the buffy coat, and erythrocytes. And they are separated by density. So you give me a second here. Draw this out for you. So here's our test tube and our three layers. Now, the majority of blood's volume is plasma. Plasma contributes to 55% of overall blood volume. The other 45% is due to what we call the formed elements. And the formed elements, as I mentioned, give us the other 45%. Of this 45%, my 1% here, this tiny little area, is referred to as the buffy coat. And it contributes to less than 1% of the overall volume of blood. Included in the buffy coat are our leukocytes and our platelets. The other 44% of our formed elements, so all of this here, are erythrocytes. So, the overall volume of blood is divided up between plasma at 55%, our leukocytes and platelets at 1%, and erythrocytes at 44%. The erythrocytes and the leukocytes and platelets together create the formed elements, which create 45% of the overall volume of blood. When we look at the formed elements by themselves, this 45%, we can see that the erythrocytes make up the majority of the formed elements. So I can color these in for you guys so you know. Oops. So there are your erythrocytes. And then we will make the plasma. Let's do a yellowish color. And we'll keep the buffy coat white because it contains those leukocytes, those white blood cells. I'd like to help students remember it this way. If you are old enough to remember Buffy the Vampire Slayer, she would kill bad guys. So the buffy coat, like Buffy the Vampire Slayer, contains leukocytes, and leukocytes kill bad guys. They kill pathogens. So we're going to come back and forth to this image here a couple of times throughout the PowerPoint. Okay, we're going to focus on plasma first. Plasma is a straw-colored liquid, and as you can see here, the majority of it's made up of water, 92% by weight. As we mentioned, plasma contributes to 55% of the overall blood volume, and it contains some proteins listed here: albumin, globulin, fibrinogen, and regulatory proteins, and we're going to talk about those individually. And then a very small percentage of plasma, 1% by weight, is composed of other dissolved solutes, things dissolved in the water like electrolytes, nutrients, gases, and waste products. So let's go back to this picture here and fill it in a little bit more. So we said that water, H2O, contributes to 92% by weight. Just a reminder. And then we have our proteins, which are 7% by weight. And then we have our dissolved solutes. Which is less than 1% by weight. Of the proteins, we had mentioned that we have a few of them. Albumin is one, globulins is another, fibrinogen, and then regulatory proteins. So we're going to write about each of these in here as well. So we'll list them out for you. Sorry. And basically what we're building here is a cheat sheet to study with. It's going to organize a lot of the information for you, make it clear and to the point. Okay. All right, let's go back to the PowerPoint real quick. And we will start with albumin. Albumin is the most abundant plasma protein, and it's responsible for a few things, most importantly for us, regulating water. Albumin is a really, really large protein, and it's located in the blood vessels, and it's too large to diffuse out of the blood vessels. So it's a solute that stays in the blood vessels at a higher concentration. And if you guys remember going all the way back to the beginning of the semester when we talked about there being a high amount of solutes in an area, that means a low amount of water. And because there's a low amount of water there, water is always going to move towards that area, right, from high to low. So albumin being present in the circulatory system, in blood, is always making sure that we are trying to regulate water inside and outside of the blood, the circulatory system. And we'll see this come up again when we talk about the urinary system. Albumin also helps to transport things like hormones and ions throughout the body. Fibrinogen is a hormone that is responsible for blood clotting. It's a protein fiber that binds to platelets. It's kind of like a net that catches platelets and allows blood to clot. Regulatory proteins are any sort of proteins that we find in the blood that are small, things like enzymes and hormones. And the reason why they're called regulatory proteins is that both enzymes and hormones affect metabolic processes of the cell. So less than 1% of the proteins in plasma affect cellular metabolism. The last type of plasma protein is the second largest group, 37%. And we have three different types of globulins, alpha and beta, and then gamma. Gamma globulins are a fancy way of saying antibodies, so either term's acceptable. Alpha and beta globulins protect lipids. If you guys think about it, the majority of plasma is created by water, and water and lipids, we know, don't get along, right? Lipids are hydrophobic. So we can't just transport lipids in blood because the blood will repel it. So the alpha and beta globulins are these proteins that are attracted to lipids on one side but attracted to water on the other. So they kind of cover the lipids and form this cloak around them that makes them soluble in the water, meaning that we can then transport them. To help you remember the list of plasma proteins from most abundant to least abundant, which is a level 1 objective, we have a little saying down here: a good friend remembers. So A is albumin, good is globulin, friend is fibrinogen, and remembers is regulatory proteins. So we can go here, and on your chart we can fill in some more information for you. Albumin regulates water or fluid volume. Globulins include antibodies and transport lipids. Fibrinogen is used as a clotting protein. And then again, those regulatory proteins are any proteins that affect metabolic processes of the cell. So things like hormones and enzymes, anything that has the potential to change what the cell is doing. And if you notice, I've listed them from most abundant to least abundant: albumin, globulins, fibrinogen, regulatory proteins. A good friend remembers. All right, so that takes care of our plasma. Now we're going to move on to the formed elements. If you remember, if we look here, our formed elements make up 45% of the overall blood volume. And the formed elements are composed of leukocytes and platelets, which are in the buffy coat, and then erythrocytes. Erythrocytes, again, making up the majority of the formed elements, but 44% of the overall volume of blood. So we are going to start by looking at erythrocytes, then we're going to look at leukocytes, and then lastly platelets. Now the first major vocabulary word I want you to learn is hematocrit. When you go to the doctors, a lot of times what they'll do is take a hematocrit number or percentage, and it's basically the percentage of the erythrocytes are contributing to the overall volume of blood. And if you remember, going back to this picture here, according to our picture, the hematocrit number would be 44%, because erythrocytes contribute, on average, 44% to overall blood volume. But that doesn't mean it's always the case. Men, for example, tend to be between the ranges of 42 and 56%, while females are between 38 and 46% for their overall erythrocyte contribution to blood volume. The reason for this is that females menstruate every month, so they are losing blood, including erythrocytes, so their overall hematocrit number or percentage of RBCs is lower. Now, erythrocytes, or often known as red blood cells, right? Cyte meaning cell, is kind of a misnomer. It's not a properly named structure, because in order to be a cell, you need to have a nucleus, a cell membrane, and a cytoplasm. But erythrocytes are lacking a nucleus and organelles. Why do we call them red blood cells? Well, somebody decided that, and we have to stick to it. Now, the overall function of the erythrocyte is to transport the respiratory gases, and they do this with the help of a structure known as hemoglobin. Within each erythrocyte, there's about 280 million molecules of hemoglobin. And the hemoglobin's job is to transport oxygen and CO2. Now, the magic number for everything we're about to discuss next is 4. For example, hemoglobin, globin here, is created by 4 globin or globin chains, 2 alpha chains, and 2 beta chains of the protein. Now, each of these chains carries a heme group, so we have 4 heme groups. And each heme group contains an iron molecule. So we have 4 iron molecules. Why is iron so important? Well, iron is necessary to bind to oxygen. So if we have 4 iron molecules, that means that each hemoglobin can bind to 4 oxygen molecules. Some of you may have heard of the term anemia. Anemia just simply refers to the inability to carry oxygen sufficiently for your body. And it can be caused by a couple of different things. One of them is iron deficiency anemia. Again, iron is needed for oxygen to bind to hemoglobin. So if you don't have enough iron in your system, that means the oxygen can't bind to the hemoglobin. So some of you may have been told that you are iron deficient if you ever donated blood. It's an easy fix. You just got to eat a little bit more iron and you'll be good to go. So overall, again, we have 4 chains, 4 protein chains creating 1 hemoglobin molecule. Those 4 chains contain 4 heme groups, and each heme group contains 1 iron, so that means we have 4 irons. And each of those 4 irons can bind to 1 oxygen molecule each, so that gives us 4 oxygens per hemoglobin. Now, because erythrocytes do not have a nucleus, that means they're not able to repair themselves. They can't undergo protein synthesis to fix hemoglobin or their cell membrane. So on average, they last about 120 days before we break them down and recycle their parts. As you already know, erythrocytes are born in red bone marrow. As subadults, as kids, this red bone marrow exists in the epiphyses of long bones as well as the diaphyses, and in all of our flat bones. As adults, it's the proximal epiphyses of some of our long bones as well as our flat bones. The red bone marrow in the diaphyses gets replaced by yellow bone marrow. Once the erythrocytes are born, they're going to travel doing their job in the circulatory system for about 120 days, picking up oxygen, dropping off oxygen, picking up CO2, dropping off CO2. And then eventually, towards the end of their 120 days, they're going to get broken down by other organs in the body and recycled. The entire breakdown process starts in the spleen. So we'll see these macrophages, these big cells that eat things, breaking down RBCs initially, and then these red blood cells or erythrocytes are sent to the liver from the spleen. In the liver, we take that heme and it's converted into something called bilirubin, which will eventually become bile. Bile is this green stuff produced by your liver that helps you to break down fats in the digestive system. The iron is carried by a protein called ferritin back to the bone marrow to be reused in the creation of new erythrocytes. And then any sort of proteins that were in the erythrocyte's membrane are broken down into free amino acids, and as you know, we use amino acids to build other proteins in the body. Now, I want to talk a little bit about blood typing. There are multiple what we call blood groups, but the most common one referred to is the ABO blood group. So I'm going to kind of jump off the PowerPoint here and move to a new picture and discuss this with you. Alright, some of you may know your blood type. We have type A blood. We have type B blood. We also have type O blood. And then lastly, we have type AB blood. What the heck does all this mean? Well, type A, type B, type O, and type AB are what we know as our phenotype. If you guys remember back in biology, our phenotype is the physical manifestation of our genes. So if we talk about a phenotype, we should probably talk about genotypes. For every gene, we actually have two copies. One gene inherited by mom and one gene inherited from our dad. If you are type A blood, then that means you inherited possibly a dominant A gene from mom as well as a dominant A gene from dad. It's usually written IA, so don't let the I confuse you, okay? You may have also inherited a dominant I allele or gene from mom or dad and a recessive one. Going back to your Gen Bio days, if you guys remember the difference between dominant and recessive genes, a dominant gene always overpowers a recessive. So in this case, as long as one of your parents gave you the gene for type A blood, the recessive gene, shown here as a little I, doesn't matter. So you can be what's called homozygous dominant for type A blood, or you can be heterozygous. Homozygous dominant means the same dominant allele, homozygous, dominant, same dominant allele. Heterozygous means you have two different genes, or what we call alleles, versions of genes, a dominant one and a recessive one. If you are type B blood, you may have received a dominant allele from mom and from dad. So again, you would be homozygous dominant, or you could have received a dominant allele from mom or dad and a recessive, making you heterozygous. But again, that dominant allele will mean that you are type B blood. If you are type O blood, that means that you inherited two recessive genes from your parents, making you homozygous recessive. So two recessive alleles make you type O blood. Lastly, if you are type AB blood, that means you inherited a dominant A gene from mom or dad and a dominant B gene from mom or dad. And both of them are expressed, and we call this codominant. Codominant. So again, going back to Gen Bio here, if you guys remember, genes are used to create proteins. So if you inherited an A gene from mom or dad, the protein you will create is called an A antigen. And this protein will be found on the cell membrane of the erythrocyte. If you inherited a B gene from mom or dad, you will have the B antigen on your cell membrane, the B protein. If you are type O, you will have no antigens, so no proteins of any shape will be produced and on your cell membrane. And if you are type AB blood, you will have both A and B antigens. So what does this look like? Well, if I were to draw a red blood cell here for my type A blood, on it would have proteins of a certain shape. And these would be our A antigens. So antigen is just a surface protein found on our red blood cells. Our B cells may have a surface protein that looks more like this. Again, our type O blood will have red blood cells that have no antigens or surface proteins on them. And then type AB erythrocyte will have both A and B proteins on them. So why are these surface proteins or antigens important? Well, in our plasma, if you remember, we have gamma globulins or antibodies. And we want to make sure that the antibodies we have in us do not get along well with the antigen we have, because if they are a match, what will happen is the antibody will bind to the antigen and then break down the red blood cell, and that can cause clotting, which is not good. So if we are type A blood and we have the A antigen, then we want to make sure that the antibody we have is the opposite of that antigen. So it's going to be the anti-B antibody. And that tells you that this antibody is against B blood, anti-B antibody. So we have an A antigen and anti-B antibodies. And if you notice, the shape of this antibody will not match the A antigen. So they will not bind to each other in your blood and cause clotting. In type B blood, we're going to have anti-A antibodies. And again, the shape of the antibody will not fit the antigen. And again, the name tells you that it is against A antigens, not B antigens. Now, because type O has no antigens, that means we can have both A and B antibodies, because there is nothing for these antigens, or excuse me, these antibodies to interact with. So we're going to have both of them here, because there's nothing on the surface of the erythrocyte for those antibodies to bind to. Now, type AB blood has both antigen A and B, which means that we can't have antibodies here. Because if we did, if we had anti-B antibodies, they would bind to the B antigen, and if we had anti-A antibodies, they'd bind to the A antigen. So let's review. Our phenotype is the physical manifestation of our genes. So if you were type A blood, that means the protein that we have is antigen A. And this is determined by either being homozygous dominant, having two dominant genes for type A blood, or one dominant and one recessive gene for type A blood. And if we have A antigen, we have anti-B antibodies. Again, we don't want anti-A antibodies in the blood because they will bind to the antigen and then cause the red blood cell to break down. Type B blood is determined by the presence of the B antigen, which is determined by the presence of the B gene or B allele. So we could be homozygous dominant and have two copies of that B gene, or heterozygous and only have one copy. Type B blood has anti-A antibodies. Again, we don't want anti-B antibodies because these guys will attach here and then cause the red blood cell to break down. Type O blood means that we don't have any antigens or surface proteins on our red blood cell. And then the only way this can be is if we are homozygous recessive. And because we don't have any antigens, that means we can have both antibodies because there's nothing for them to grab onto. This is different than type AB blood because you inherit one gene for your A protein and one gene for B protein or antigen. So because you have both of these antigens, we can't have either of the antibodies because they will bind to those antigens. So what does all this mean? Well, if I have, let's see here, a red blood cell with A antigens on it, and there's somebody who is type B blood needing a transfusion, let's look and see what would happen here. When you transfuse blood, so as a donor, when I give my blood, the only thing the recipient is receiving is the erythrocyte. Nothing else comes with it. By the time that blood will make it to the recipient, it's just the RBCs. Okay? So I've just drawn a red blood cell here, a type A red blood cell, because that's the only thing this B person is receiving. But in the plasma of this B person, we have anti-A antibodies. Now, we know the anti-A antibodies won't attack the B antigen, but when I donate the red blood cell that has A antigens to the person with B blood, what's going to happen is this anti-A antibody will find the A antigen and bind to it, causing the red blood cell to break down and coagulation or clotting to occur. And this can lead to cells dying and then overall organ failure. So let's do another one. I got a new sheet here for you guys. So let's do it again. Let's say I have somebody who is type O blood, and we know type O, if we go back here, means it has no surface antigens, and it has both A and B antibodies, but because it's the donor, we're only concerned about the red blood cell. And let's say this person is donating to somebody who is type A. So we know type A blood has A antigens. And if we go back here, we see type A antigens and anti-B antibodies. So let's go ahead and draw those in. Now, if we were to donate these type O red blood cells to somebody who has type A blood, would anything happen? The answer is no, because these anti-B antibodies only bind to the B antigen. Type O blood doesn't have any antigens, meaning these antibodies won't bother it, and we will have a successful transfusion. So that brings me back here. If we look at these two blood types, type O is known as the universal donor because it gets along with everybody. Type AB blood, on the other hand, is the universal recipient, meaning it will always be able to take any sort of erythrocyte because it doesn't have antibodies. It will get along with everybody. I'll show you. So let's do another example. Let's say, let's say this is my donor and it's AB, so it has antigen A on its surface as well as antigen B. And let's say we are donating type A blood. Again, our donor, we're only concerned with the red blood cell. So I'm only drawing the red blood cell and the antigens. Type AB blood, if we look back here, we know it has both A and B antigens and no antibodies, so I don't have to draw any antibodies, which means that when this red blood cell is introduced to this blood, there are no antibodies here that will ever attack the A antigen. So because we have no antibodies in AB blood, it's always accepting of any sort of antigen. Now, where things get tricky, and where some of you may be asking some questions, is our last thing we need to consider, which is RH. RH is what determines if we're positive or negative. So some people you may hear them say, well, I'm A positive or B negative. RH is the presence or absence of a protein called the Rh protein or the rhesus protein. If you are homozygous dominant for Rh, so again, that's homozygous dominant, or heterozygous, it means that you are positive. It means that you have the protein. If you are homozygous recessive for Rh, that means you do not have the protein and you are considered negative. Some of you may be curious as to how we write Rh. So when we look at our genotype, we have that I again like we saw in the other blood types. But we, instead of writing Rh, we use the letter D. It's just the letter designated to talk about Rh. Now, if you have the Rh protein, which we call the Rh antigen, then you have no antibodies. If the Rh antigen is absent, meaning you are Rh negative, you also have no antibodies, unless you are exposed to the Rh antigen. So for all the other blood types, we're born with antigens and antibodies. But for Rh, we are only born either with the antigen or without it. None of us are born with the antibody for it. So let's talk about how you do produce the antibodies. Let's say we have a red blood cell here that is A positive. Now, I know type A blood has the A antigen, so I'm going to put those on there. And I said A positive, so that means we have the Rh protein. So I'm just going to draw little pluses on here because we have the Rh protein, the Rh antigen. And let's say I want to donate to somebody who is AB negative. So I have A and B antigens because I'm AB. But because I'm negative, I don't have these Rh proteins. Again, when we donate, we're only donating the RBC. And in the recipient's blood, because it's AB, we don't have any antibodies. So what happens when we donate this blood? Well, we know that the A antigen will not be attacked because we don't have any A antibodies in this blood. But this Rh protein, this Rh antigen, is a new foreign substance for the blood. It's never seen this before. So upon first exposure, the body will create antibodies for Rh. So upon first exposure, we'll create antibodies. The patient may be slightly sick, but they would most likely survive. If we introduce that blood again, if you notice, our blood is already primed and prepped with antibodies for the Rh antigen. So upon second exposure, these antibodies, these Rh antibodies, will bind to the Rh protein of this A positive blood, causing that agglutination or that coagulation, that clotting of blood. Blood groups here. Let's move on to leukocytes. So leukocyte is another term for white blood cell, and these are true cells. They contain a nucleus and organelles, but no hemoglobin, so they don't transport gases. They are involved in immune responses and help to defend us against pathogens. They're usually slightly larger than RBCs or erythrocytes, and they are able to move around, at least some of them, using blood and exit the bloodstream when needed via a process called diapedesis. And the reason they are able to leave a blood vessel in a certain area, or why they would want to, is because they are attracted to that area by chemicals released by damaged cells. And that releasing of chemicals from the damaged cells is known as chemotaxis. So let's look at this. Let's say that you were pricked by a pin, and you killed some of the epithelial cells and some of the cells in the dermis. And with that pin, we had bacteria brought in. Well, these dead or dying cells are going to release chemical signals that attract white blood cells to the area. That process is called chemotaxis. We're sending a chemical signal. Now, once the white blood cells are near that area, they're going to squeeze through the endothelial cells of the blood vessels, and we call that diapedesis, to move into the area and eat away the bacteria and damaged cells. So these are phagocytic cells, they're eating cells. Now, we classify leukocytes based on what we see underneath the microscope. So they're divided into two classes, granulocytes and agranulocytes. Granulocytes are white blood cells or leukocytes that we are able to see their organelles underneath the microscope, and they show up in these little granules inside of the cell cytoplasm. Agranulocytes have organelles in them, but we can't see them underneath the microscope, so we call them agranulocytes, or without granules. If you look here, we have three types of each. We have three granulocytes, neutrophils, basophils, and eosinophils, and then two agranulocytes, lymphocytes and monocytes. So three granulocytes and two agranulocytes. I always tell students to associate Phil with gran. I always say grandpa Phil. And all of the agranulocytes end in the term cyte, so that kind of helps you organize them. So what we're going to do is go through and be very, very straightforward about each of these. Do not try to learn more about them than what we do in this lecture. So first and foremost, one of our agranulocytes is called a neutrophil. If I were to ask you about the anatomy of a neutrophil, I would expect you to say that it's multi-lobed nucleus. So here, this is a nucleus and it has many lobes to it. Obviously that you can see the granules because it's a granulocyte. And that those granules are a pale purple color. So again, the anatomy of a neutrophil, it's multi-lobed nucleus. It has a multi-lobed nucleus and it contains pale purple granules. Now, as far as function is concerned, it is a phagocytic type cell and it eats bacteria. That's all I want you to know for the function. And then I would like for you to write down or make a note that they are the most abundant leukocyte that we have, most abundant white blood cell that we have. The next type of granulocyte is called an eosinophil. If I were to ask you about the anatomy of an eosinophil, you would tell me that it is bilobed as far as its nucleus is concerned. It's got two lobes, and that its granules are a red color. This is why it gets its name eosin. Eosin is a red dye that is attracted to the granules in the eosinophil. Now, as far as the function goes, it is a phagocytic cell, and it eats pathogens that have antibodies attached to it. And we call this an antigen-antibody complex. So essentially, an antibody will go out and search for a pathogen, bind to it, and then that signals the eosinophil to eat it. It also helps to destroy parasitic worms. This is the second least abundant leukocyte. The last type of granulocyte is a basophil, B-A-S-O-P-H-I-L, basophil. And what the anatomy of this cell looks like is it's a rather large cell, and it has so many dark purple granules, you can't even really see the nucleus. So all you see here is just a cytoplasm filled with these dark purple granules. This guy secretes something called histamine, and histamine causes swelling. And it's usually released during an allergic reaction, which is why you get that congestion and that swelling occurring. And if any of you have had an allergic reaction, you know that you take antihistamines to reduce the inflammation. So basophils are responsible for releasing histamines. These are the least common of all of the leukocytes. Now, onto our agranulocytes. Our lymphocyte would be described as having a large nucleus with a small border or halo of cytoplasm around it. Because it's an agranulocyte, we will not be able to see granules in the cytoplasm. The function of lymphocytes is to attack infected cells and create antibodies. The other major function of lymphocytes is to initiate an immune response. So attack infected cells, initiate an immune response, and produce antibodies or gamma globulins. These are the second most abundant of all the leukocytes. The last agranulocyte is a monocyte, and monocytes are really large, and they have what we call a kidney bean-shaped nucleus. Kidney bean-shaped nucleus. These are the cells that we were looking at earlier that are able to squeeze between the the endothelial cells via the process of diapetesis and eat that bacteria. So these are phagocytic cells. You can see here exit blood vessels and phagocytized pathogens. So eat dead cells or bacteria. These guys are right in the middle. They are not the most abundant or the least abundant, but right in the middle. So to summarize, we never let monkeys eat bananas. Neutrophils are the most abundant. Lymphocytes are next, followed by monocytes right in the middle, eosinophils, and then basophils, from most to least abundant. The last thing we see in blood, and the last one of our formed elements, so to go back here, we talked about erythrocytes. We just got done talking about leukocytes. The last formed element is a platelet, and that's in the buffy coat. Platelets are basically protein-type cellular fragments that are very, very small in comparison to red blood cells, about a quarter of the size. And when needed, they will bind to that fibrinogen, that dissolved protein in plasma, to clot blood. And that's really all you need to know about them. So, if you'd like, you can go ahead and test your level 1 objectives here. List the functions of blood. Erythrocytes contribute to blank % of the overall volume of blood. Go ahead and, if you'd like, pause this video and give yourself an opportunity to look at the PowerPoint you have at home and answer these, and then I will reveal the answers. So go ahead and pause now if you want to take a moment, and then press play when you're ready to hear the answers. All right, list the functions of blood. We know that blood is responsible for transportation of things like gases, waste, nutrients, enzymes, proteins. We know that it's there to regulate things like pH, temperature, and fluid volume, as well as an immune or a protection due to immune responses and the leukocytes in blood. Erythrocytes contribute to blank % of the overall volume of blood, and that would be 44%, which is not a protein found in plasma. That would be A, globin. List the 3 formed elements. They are erythrocytes, leukocytes, and platelets, and those make up 45% of the overall volume of blood. Blank is the process of signaling leukocytes to an injured area via chemicals. That is chemotaxis, B. Blank are lacking visible granules in their cytoplasm. That would be agranulocytes. And then list the 5 types of leukocytes. In order from most abundant to least abundant, we have neutrophils, lymphocytes, monocytes, eosinophils, and basophils. All right, if you guys have any questions, please feel free to ask or send emails over the weekend, or try to catch me before lecture. If you want to come in a little early, we can go over some of this.